So in our Mycology and Microbiology Center, we just generated also a Twitter account and a YouTube channel in which we upload at least most of the presentations. Well, let's say those ones that are not embarrassing. So let's continue. In this talk, I will focus on high-throughput sequencing and especially long-read sequencing benefits. I will also talk about differences among various high-throughput sequencing methods, including both second and third generation ones, and also hints what to prefer on specific occasions with specific objectives. High-throughput sequencing has greatly benefited our understanding about microbial diversity, and in some cases also about the ecology of macroorganisms, such as plants and small animals. In essence, high-throughput sequencing is a method of parallel sequencing of individual DNA molecules. Essentially, it is replacing cloning, which was followed by Sanger sequencing. But as you might remember, cloning was a very tedious and costly process. Furthermore, high-throughput sequencing enables parallel analysis of hundreds or even thousands of samples by using specific primer attached DNA barcodes. High-throughput sequencing, since its beginning, has improved the throughput of all data by tenfold to nearly 1 billion fold. And it also includes 1,000 to 1 million fold improvement in the genome analysis in terms of cost and time. On the graph, in the graph on the right, you will see the development of various high-throughput sequencing techniques by year, and on the y-axis you also see the output in terms of megabases of data. Now we have almost linearly moved on in the last 6 years. What is also important about all different high-throughput sequencing methods, and especially when it is amplicon based, is that the PCR has to be performed using tagged primers, which have sample-specific indexes attached, or they may also additionally have sequencing adapters attached, so that these primers generate indexed amplicons. Also, of course, it's possible to analyze amplicon-free DNA and also RNA fragments using more specific techniques. Long reads have become available in the last 8 or 9 years, and their main benefits include much greater taxonomic resolution. Related to this, also much better phylogenetic information. And when it comes to genomics or metagenomics or functions in general, then substantially longer genomic or metagenomic contigs, which are beneficial for separating alleles, isoforms, and phases, which is different or mapping different haplotypes in the genome. When it comes to taxonomic resolution, together with Sten Anslan, we recently prepared a graph illustrating the differences in sequence similarity and number of absolute differences in short and long barcodes. So in the graphs, on the left graphs, there is information about bacteria. On the right graphs, there is information about fungi. The top graphs indicate absolute sequence similarity, which indicates that in both bacteria and fungi, the ITS region is most variable, but when it comes to number of absolute differences, then those long reads that are comprised of both ITS region and LSU and SSU, these display much greater overall variability and characters to distinguish those organisms. Just to give a brief overview about the methods. First, second generation sequencing methods. Here I'm comparing Ion Torrent, Illumina, and Chinese MGI tech methods. You might also recall that once upon a time there was 454 sequencing. This has become obsolete because of relatively low sequence quality and high cost. Nowadays, this technology or similar technology is used by Thermo Fisher's Ion Torrent sequencing. So it's principally quite analogous to what was 454, including the technology itself and error rates. So in principle, in Ion Torrent, amplicons are ligated to beads. Then there will be carried out emulsion PCR so that the amplicons in beads are replicated. Sequencing is performed by synthesis by reading release of protons. Incorporation of different nucleotides releases a different amount of protons, and this is recorded by the machine. So Ion Torrent machines are such kind of medium-size benchtop machines. And the best available chips include 530 and 550. More about these later. The next and perhaps most broadly used technique nowadays is Illumina sequencing by Illumina Incorporated. The known platforms include MySeq, HiSeq, and NovaSeq. Here, the amplicons are first linked to tiny glass plates, and on those plates there is performed so-called bridge amplification, where those single molecules are replicated to thousands of different molecules, and then again the sequence reading is done by synthesis when polymerase adds one or the other of the four nucleotides. then the fluorophore is released, and each of the nucleotides is associated with a different fluorophore. And sequencing is based on recording the fluorescence across time. And the newer Chinese MGI technology is somewhat similar to Illumina in the end phase, but in the early phase, it is based on a circularized molecule library, which is achieved by rolling circle amplification and formation of DNA nanoballs. Those nanoballs are then sequenced by ligation using ligase enzyme, and recording of the sequence is again done by recording the fluorescence. The equipment, the newest, most high throughput equipment for Illumina and MGI tech are big shelf-like machines, which are quite big and heavy. Now, third generation sequencing methods. There are two of these. First of them is Oxford nanopore technologies, which was actually developed later than PacBio. Oxford nanopore has 4 different sequencers, which very strongly differ in size and throughput. To name the major ones are the Flongle, MinION, or the main one, GridION, and PromethION. Oxford nanopore or nanopore sequencing is very flexible. It allows also direct sequencing of RNA, but usually still DNA molecules are sequenced. So the ordinary PCR products are merged into a specific DNA library, which needs specific preparation. It includes other short nucleotide elements and different motor molecules that, when put into the machine, lead the construct or DNA molecule through the nanopore. And reading of the sequence comes in pentameres. The sequencing signal in the machine looks like this. So there are specific formula how to understand those combinations of pentameres. And also, whenever there are methylated bases or any other types of modified bases, these produce different signals. So there is still probably a lot of optimization to do to recognize all these signals and remove this extra noise to get higher quality sequences. So the high error rate, 5%, sometimes also reported as little as 3%, is still too much to do common metabarcoding or barcoding with nanopore techniques. So the benefits of this method are extremely long reads. The average size is 50 kilobases in very experienced labs, and in case of very high quality DNA, and in literature there are reports of up to 2.3 megabase reads. Nanopore also supports direct RNA sequencing, for which there are separate kits. It also supports epigenome sequencing or detecting those methylation and modification to bases. It also enables detection of other artificial bases, such as bromides or bromo group linked deoxyribonucleotides. It allows very rapid diagnostics because the sequencing time may be from 10 minutes, but it may also last to several days to get higher throughput. And also importantly, it can be performed in non-laboratory conditions, such as rainforest, tropical, rainforest, Antarctica station, International Space Station, etc. And the pocket-size sequencer Minion is also quite cheap. It's around 900 euros for the small equipment. However, the library preparation is quite costly, so end users have to consider that this is one of the most expensive steps in nanopore sequencing. The second, third generation sequencing method is PacBio sequencing, provided by Pacific Biosciences Incorporated, and the most novel platform is Sequel 2. The library preparation is also very specific. It's called the SmartBell library, in which again the library is prepared by ligating the ends of the molecule and generating a circularized molecule. This circular molecule is then replicated using rolling circle amplification, so that there will be multiple tens or even hundreds of tandem repeats of the same molecule. And this trick actually adds to the very high quality consensus sequence across all those repeats. The signal recording is using a laser in a small nanowells in plates. So in each small plates there are millions of those different tubes, and the signal is recorded by the laser. Again, the sequencing is by recording the synthesized molecules, and each of the nucleotides releases a different fluorescent molecule. And those molecules have different signal times and signal intensities, but again, these can be disrupted when modified bases are incorporated. Thanks to this, PacBio can also efficiently read methylations. The PacBio Sequel 2 machine looks like giga-sized coffee machine. So it also fills one corner in the room. The main benefits of PacBio are also reasonably long reads. The average can reach 30 kilobases. A maximum read length reach 200 kilobases. Because of recording methylations, it can also be used for epigenome sequencing, like nanopore. Although the raw error rate is quite high, 5% or even more, up to 10%, the circular consensus enables to build very high accuracy, 99.9% at 10 times coverage, and much more at 20 times or more coverage. Again, the downside is relatively costly library preparation. There are also additional approaches that utilize short reads for building synthetic long reads. So these methods are called synthetic long read methods, or UMI related methods. UMI stands for a unique molecular identifier, which is a key in these methods. For generating synthetic long reads, there is a specific PCR, and especially the primers are comprised of multiple elements. One is the ordinary primer that anneals to the template. Then there is this unique molecular identifier. There is also a sample-specific index, and also there can be a specific adapter. So the process is that there is a long PCR with those multiple tagged primers, and then there is internal ligation of those multi-tags, followed by site-specific fragmentation and short read sequencing. And later those reads are long reads are regenerated by bioinformatics tools, and later also samples are secondarily regenerated by sample-specific indexes. These methods are restricted to the length of PCR, which is from 3 to 10 kilobases. The error rate is very low because of multiple coverage of the same individual molecule. However, the throughput is greatly reduced because of this multiple coverage, and also for securing that all reads are sufficiently covered. Also, the library preparation is relatively costly compared to ordinary short reads, but comparable to the long reads. So the principal or most common method for this nowadays is loop-seq sequencing, and this slide is based on the loop-seq methodology. Alternatively, such unique molecular identifier-based PCR products can be sequenced using long read platforms the positive side is that the tedious ligation and fragmentation steps are not needed, which makes it cheaper. Again, the error rate is comparably low, and the throughput is reduced, but less. But still, compared to the deepest sequencing short reads, the throughput per molecule of those synthetic long reads is less when using long read sequencing, at least based on the present features of the sequencers. So there are quite many different methods, sequencing methods, and also different platforms within those methods. So the picture is quite crazy. And furthermore, for different approaches such as metabarcoding and metagenomics or genomics, different machines are preferable. So when we start with metabarcoding, as mentioned, the most commonly used technique is Illumina MySec 2x300 paired end. It's reasonably cheap, but the throughput has become quite low compared to newly emerged machines. And also the sequence length of up to 550 bases for the fragment is limiting. So this fragment length is actually limiting for all short read sequencing platforms. The ion torrent machine also enables up to 600 base reads, but the accuracy is still below the standards. The Chinese machine has a maximum capacity to provide up to 400 bases. So this 400 base reads. So this might be suitable for the shortest reads of bacteria or mini barcodes of plants or animals. The read accuracy is comparable to Illumina. The sequencing cost is per read approximately 5 times smaller compared with MySec, but the library preparation is several times higher. So it depends how many libraries and what kind of sequencing depth you wish to have. For PacBio, library prepping is one of the most costly compared to other platforms. The Sequel 2 has also much improved sequencing capacity, which exceeds 8 times the previous model. So it's 4 million sequences per run, and the average raw read length is 30 kilobases, which is sufficient to cover 3 kb amplicons at 10 times coverage to secure very high quality. Oxford nanopore methods are unsuitable for metabarcoding because of the low read accuracy, but when combined with UMIs, they may be competitive on some occasions. although there are no published meta barcoding studies using UMIs and nanopore combinations. Here again, if you want to have long barcodes, then synthetic long reads combined with Illumina or MGI text sequencing might also perform pretty well. When it comes to metagenomics, those long reads might be actually even more beneficial compared to metabarcoding, because in this way it's possible to construct more efficiently the entire genomes or chromosomes of microorganisms. Here the standard is Illumina Novasec 2 times 250 paired end sequencing, which is considered very cheap. It's quite accurate, but the downside is the average read length of 250 bases. So in some cases, it may not be sufficient to predict the function of a predicted protein. Ion Torrent here it has another more powerful chip, P550 chip, but the accuracy of this is still lower, although the throughput is very high. The Chinese have also a more powerful version, T7, which has analogous 2 times 150 paired end sequencing, as does Illumina Novasec. When directly comparing prices, it appears to be almost exactly twice as cheap compared to Novasec. PacBio is very strongly limited here because of the library preparation costs, because in metagenomics, essentially each sample has to be generated into different library. Nanopore methods again perform poorly because of the low read accuracy, and here synthetic long reads might also offer a good solution to produce long and accurate sequences. And so do also long reads combined with UMIs. For both of these last technologies, there are several recent papers from 2020 and from the ongoing year. So here I've compiled a list of methods and a list of platforms and the potential applicability when considering accuracy, sequencing length, the quality, and also cost. But of course, it depends on the budget and individual research objectives of those different projects. So to start this kind of rapid diagnostics within a few hours is so far only principally possible or feasible using Oxford nanopore technologies, either Flongle or Minion. Metagenomes and transcriptomes for short reads, less than 500 bases, are certainly most feasible using Illumina or MGI tech. And so is metabarcoding of short barcodes or those mini barcodes. When it comes to medium length metagenomes, transcriptomes, and metabarcodes from 0.5 to 4 kilobases, then it seems that PacBio without UMI is performing best, because up to 3-4 kilobases reads you can get sufficient consensus sequence coverage. For longer fragments from 5 to 10 kb, both metagenomes and metabarcoding, synthetic long reads are probably the best option, or alternatively UMI combined with Oxford Nanopore and PacBio are also performing well. There is also a hybrid approach which combines single short reads and then either synthetic long reads or natural third generation sequencing long reads. So these are also quite promising for especially metagenomes and genome analysis. But these are a bit more costly because one needs to run it several times using different platforms. For detecting alleles, isoforms, and haplotype phases, again, it seems that this kind of hybrid approach between short read and long read sequencing seems to be the best. To sequence epigenome, principally the only methods that do it in a straightforward manner are PacBio and Oxford Nanopore. Here, this UMI-based approach is not applicable because methylations become lost during PCR. Which of them is better? I don't know. Some say that Oxford Nanopore is more sensitive when recovering various methylations, but PacBio might be more accurate because the methylations again can be recognized in several consensus reads, and so there can be some kind of statistics behind. Direct RNA sequencing is at present possible only with Oxford Nanopore technologies. So that's it. Thank you. Do we have questions? I hope you do. Wakey, wakey.
I have one if I can ask.
Yes, you can.
Thank you. So imagine field work. You go with small lab, you have microscope, and you are not working in ecology, but you work with, let's say, out ecology with specific species. And you take either maybe root samples, some soil samples, or other small organisms or pieces of organisms. So did I get right that the best way to work in the forest, in the field, is to have this Oxford Nanopore with you, and you take samples and fast analyzing, rapid identification to know that you have right species or microorganism. Is this the way with nanopore to go?
Yes, this is one possible way. Of course, it is a very costly way of just identifying a species. But if you have restrictions, like you may not carry the material out of the country or some other similar reasons, it's indeed one of the options. But you also need to think about how you are going to extract the DNA in the field hut, if this is possible. So
Yeah.
it's probably not possible from soil, but it's certainly possible from fungal or plant tissue.
Yes, this was my point, that if you want to bring back to lab specific species alive, then this might be, especially from wood or some mycelium, or so, to know that you have correct tissue with you. It is expensive, of course, but if you need specific species, I think this, and you do this just maybe few species per year actually to get them. Otherwise, you come back to lab, wait for a few days to get result. Okay, it's not that, and going back to the field to pick it up. So this was my, when you are hunting specific species to get alive back to lab.
Yes, that's the way probably.
Thank you.
A question, and can you hear me?
Yep.
Yes.
Yeah, and a comment. So for your previous answer to Urmas, actually you can extract DNA from environmental samples as well from in the field. There are some new kits and you can easily do this. But I had a question about those UMIs. So have you actually seen some studies that, or metabarcoding studies that use UMIs for capturing longer metabarcodes?
For metabarcodes, I would actually say no, because most of these have targeted metagenomes. But as far as I know, there are certainly people using those UMIs related to this loopseq technology.
But still mostly for genomics, metagenomics.
No, they have developed also specific kits for 18S analysis of bacteria, and also there was something for fungi, which included 18S and ITS. So, sorry, 16S for bacteria.
Okay, and benefit would be higher throughput, right? Compared with, for example, BacBio. You still get long synthetic reads, but then the throughput, because you're sequencing with Illumina, the output is still higher.
If you use this ultra high-put Illumina machine, your throughput will be still about 5 to 10 times higher.
Okay, and all in your graphs, all of those UMI or long synthetic read stuff for metabarcoding and all those suggestions were not based on any like references, the studies that have been actually made, but just you thought it might work well.
Well, there are studies, but these studies are not yet published.
Yeah, okay. So for me, there are no studies.
Unfortunately, yes.
Okay. Yeah, cool, thanks.
I have a small note to Sten. Sten, can you send me kits you
Yes.
noted?
Okay.
Okay, thank you.
Yeah.
Thank you very much for the presentation. I also have one question about metabarcoding and using UMIs. Would you think in like a future perspective, would the Oxford nanopore be a more suitable method compared to loopsec if the accuracy improves?
That's a very good question, because we also have to assume that accuracy of loopsec or the throughput of loopsec will improve. And also the prices become less for both of the methods. So it really depends on the future. But if we consider that nanopore accuracy might grow to 99% in the next 5 years, then I believe that nanopore is actually the future. Not necessarily Oxford nanopore technology, but some kind of nanopore technology, because there are alternatives as well.
Okay, thank you.
Rain is here. Can I ask, please?
Of course.
Yes.
So what do you recommend for population genetics? Which kind of methods you would like to use in future?
How much money do you have?
As you know, not very much.
Then don't do it.
Okay.
Well, in principle, I would probably recommend going for this genome skimming, which is a very low coverage sequencing of the genome, and then using some very short read method. If you want, however, to do some kind of very high quality population genetics or genomic studies, such as the Estonian Biocenter is doing for humans, then of course I would recommend long read sequencing, perhaps using PacBio, because now even those American sequencing centers, they say that they are doing everything in human genomes using PacBio. So Illumina also has become suboptimal, let's say.
Oh, thank you. This is nice to know. PacBio is more expensive.
Yes, but you can consider that for an average fungal genome, you can probably get about 20 genomes sequenced, but you will have very high library preparation costs, because each genome or each specimen has to be prepared as a separate library.
In PacBio?
In all, but in PacBio, this is the most expensive in relative terms.
Okay, thank you.
Thank you, Leho, for useful presentation. I have also a question. I want to know how widely is this Chinese technology used by the scientific community? Probably this is understandable from the papers coming out. Is there any data, metagenomic or genomic data released using this technology or not?
Yes, there are actually quite many data available already. It is not the method of choice by the Americans and then Europeans, because there are some how to say, patent conflicts between Illumina and MGI Tech. So the American company claims that many of the MGI Tech individual technology pieces are scooped from Illumina.
I would add here that also the MGI deck is relatively new compared to Illumina, so we don't know much about it here in Europe and also in US. So West-oriented, but in China they have been using it longer time.
I think the usage is growing very rapidly. You might want to know that in Riga, Latvia, there is a big MGI Tech sequencing center. And as far as I know, they are also doing service. So they are not just sitting there. So something must be happening.
Yeah, but this T7 came out last year, right? The most powerful MGI deck machine.
It was either the last year or 1 year before.
So, and compared to Illumina, like 2011 or something like this, so it's, we don't know much about it yet.
Yes, but this T7 machine is already available in some other countries besides China as well. So mostly the information is out in preprints, where research teams want to, like, in a way, prioritize their use or show the priority of their use of some specific equipment, or just to put down some kind of landmark. In a way, these are good because we can see that whether they work or whether they are so much pushed up that it's just ridiculous. But there are actually both types of studies for this MGI Tech, and also there are such studies for MinION.